Hello viewers, welcome to Mesology. In this video, our focus is mainly on the ultrastructure of the gamete cells in human. The female gamete called the ovum and the male gamete called sperm. Let's get started with the ultrastructure of the female gamete. The ovum is a large cell in the human body, typically visible to the naked eye when mature. It measures approximately 120 micrometers. If we speak in inches, it is 0.0047 inches in diameter. The living content is called the ooplasm. The zooplasm is the yolk of the ovum, a cell substance at its center which contains its nucleus called the germinal vesicle and the nucleolus called the germinal disc. The ooplasm consists of cytoplasm of the ordinary animal cells with its spongioplasm and hyaloplasm, often referred to as the formative yolk and the nutritive yolk. This nutritive yolk, which is called the deuteroplasm as well, is made up of rounded granules of fatty and albuminoid substances embedded in the cytoplasm. The mammalian ova contains only a tiny amount of the nutritive yolk for nourishment of the embryo only in the early stages of development, but in the oviparous animals it is considerably large and present throughout. The ovum in the human are surrounded by protective as well as nutritive cells. Here, just outer to the ooplasm is the layer of perivitelline space. Following that, there is the layer of zona pellucida. And finally, towards the outside are the cells of the corona radiata. Now let us know the difference between an ovum and an egg cell. An ovum is usually an unfertilized female gamete, while an egg is its fertilized state. So, an egg will contain both maternal as well as paternal genes. The genetic material of an ovum will be haploid, while that for an egg will be diploid with its genetic material. The human ovum passes through several stages of development, starting from the primordial follicle, moving on to the primary oocyte, secondary oocyte, which passes through the stages of Graafian follicle, and finally the entirely mature ovum. Our next focus is on the male gamete, that is the sperm. A mature spermatozoon is a highly differentiated single cell that comprises of a head, neck, middle piece, and the tail regions. All these parts are bounded by a single continuous plasma membrane. An ideal sperm has a flattened, pear shaped, smooth lined head and a clearly visible and well defined acrosome present anteriorly. The acrosome is a cap like Golgi derivative immediately beneath the plasma membrane. The sperm head measures around 4 to 5 micrometers in length and around 2 to 3 micrometers in width, with the acrosome which covers approximately 40 to 60% of the anterior part. The shape of the head and its size is under tight genetic control for each species. The DNA content and the proteins that are present along with certain vacuoles is very particular for the different species in the nuclear organization. The neck has the centriole. Then it starts with the further part of the neck or the middle piece, which is composed of rigid proteins, typically 5 to 7 micrometer in length, which includes the connecting structures of the neck and the anterior part of the sperm tail. Here, flagellum or the axial filament complex is surrounded by a double spiral of elongated mitochondria. Around 12 to 13 gyres of mitochondria are present here. The distal limit of the middle piece is marked by an annulus. Then the sperm tail is the last portion. This is built upon a typical 9+2 microtubule structure of cilia and flagella, which is regulated by the hundreds of microtubules that are associated with proteins and the motor proteins which are involved in controlling the swimming direction for the sperm cells. About half of the proteins is a fibrous sheath that imparts the sperm its mobility. Thank you for watching. Please do like, share and subscribe to my channel.